We'll get going here in just a moment.
Yeah. All right. So people are flooding in. All right. So I think we should get started, and we do have a packed agenda. So all right, I'm gonna start us off. So my name is Beth Marmino. I'm from the Stanford ADRC, and I'm also one of the many co-PIs of CLARiTI. It's so exciting to be here with the ADRC community today. I just want to make a quick note that the website for CLARiTI is now live. The link is just shown up here on the title slide. So for the webinar, just make sure you're muted. Feel free to write questions in the chat as we go. We're going to have time for questions at the end as well, so you can raise your hand then. All right, Sterling, can you go to the next slide? So first, I think it's so important that we acknowledge how collaborative this project has been. We have 9 MPIs, we have over 40 co-investigators. These are all from different ADRC sites. We have interest from all ADRC sites, and we have key industry partners that are going to enable us to do widespread PET scanning across the whole ADRC network. This collaborative nature is really a defining feature of this project, and I just want to thank everybody here for being part of this team. Next slide. So as I mentioned, we have a lot that we'd like to cover today. So we're going to try to give information on all these topics listed here. We'll be sharing a recording of this presentation. We'll be sharing the slides, study documents, and also the website I mentioned. So you'll have various materials for reference. So some of these topics we might go through quite fast today, but hopefully we'll touch on all of them. All right, next slide. So we've already received a number of questions, from the community on this project. So we're going to have time for questions at the end, but we wanted to address just a couple of questions that have come up a number of times across different sites. So one question is about the single IRB, although technically it probably would be possible to do this, without a single IRB, we really would like to do this with a single IRB. This is going to make things a lot more straightforward in terms of managing the project and making sure that we're counting the data flow correctly. Another question that has to do with the P30 requirements. So this has caused a lot of confusion. And yes, so the CLARiTI enrollees are, in addition to the P30 requirements of 24 participants with ATN imaging per year. So everything we do with CLARiTI, all the CLARiTI enrollees are on top of that expectation from the P30. We've had a lot of questions about funding. Sterling is going to be going over this today. And then another common question has been about redundant procedures. So, specifically about blood and MRI, if this has already been done within the last year. So the answer to that is we won't be repeating that. You won't need to repeat that. For instance, if you have blood at NCRAD, if you're participating in diverse VCID. Another, I guess, potential flavor of this is if you've already done a scan MRI on a participant in CLARiTI, we think this would enable you to waive the MRI, but we need to confirm some data sharing details to make sure the data sharing is the same as what we are going to be doing in CLARiTI for that to be okay. All right. And then finally, so here, this is where we are now. This is a kind of a timeline. And before I hand it over to Sterling, I just want to bring up, this is something that we've been working on as a group, as a collaborative effort with all of you for the last 3 years. So this really started in 2020 through concepts that were brought up through the imaging steering core, or the imaging core steering committee. It's involved multiple back and forth via surveys and interviews with the sites. And then finally grant writing and review, and we finally now have the actual funding to do this. So I mean, it's such a privilege for me to actually see us transition from the concept phase to the actual launch of this project. I just think it's so incredibly exciting. So with that, I'm going to turn the presentation over to Sterling Johnson from Wisconsin, who's also the contact PI of this project. Sterling.
Thank you, Beth. It truly is exciting to be where we are now and to be talking with all of you today about startup for this project. We have some great news. We've submitted the IRB, the initial version of it. It's going through its first round of review, and we'll certainly continue to accept feedback from you as we go back and forth with the IRB. The IND will be going in shortly as soon as the protocol details are settled with the IRB, and we are starting in earnest to do site qualification and training. Starting in January, the site subawards will be starting to roll out, and as fast as we can get those negotiated with each of you, we can begin this study, and super excited to do that. We'll do another one of these webinars in February, and hopefully by March, we'll be able to scan our first patients into this project. The goals of this, as you all know, are to use biomarkers to provide some clarity on the multiple intersecting etiologies that may be affecting our patients and the temporal features of those etiologies that are affecting our patients. Doing all this, we want to provide resources to implement standard imaging across the center program and make generalizable discoveries through diversity and inclusion in this project. And as Beth mentioned, our goal today is to help you understand a little bit more the protocol and the funds framework and address your questions and get your feedback on where we'll go from here. So CLARiTI is integrated into the ADRC ecosystem. That's been our plan from the start. We're not trying to reinvent the wheel, but use the existing infrastructure. And that's largely through NAC. The NAC data platform will accommodate CLARiTI. We'll have a place at Lonnie where these images will be uploaded, likely using the same interface you're using for scan uploads. There may be a few differences there so that we can distinguish these images that are uploaded from the generic scan uploads. We'll be integrating with NECRAD through the blood that we're collecting, which will be leveraged with the existing blood that's there and the infrastructure there to do these assays. And while we're not planning on doing genetics per se, the blood will enable genetics on anybody who hasn't had that done, and that'll be available too. And all of these will be available to researchers everywhere through the NAC front door. And a big thank you to NAC for enabling all of this. The protocol, these materials are available to you. They're in the link in the meeting invite for this meeting. They're also going to be on the NAC website, maybe even later today or shortly after this meeting, in the next few days at least. But here's an overview of the protocol, and many of you have seen this before when we presented it in San Diego. Screening, consenting, a scan-compliant amyloid PET at baseline and approximately 24 months. For those of you who are on a 3-year schedule for PET, that's going to be allowable, but the defaults would be 24 months. Tau PET at baseline, and then conditionally at follow-up, and conditionally means if they're normal, cognitively unimpaired, and their prior amyloid scan and their tau scan were both quite negative, there's no need to redo their tau scan. The scan MRI will be our base protocol, and to that we'll add a few other a few other additional sequences that John Detra and others are working on, and we'll be talking about that at an upcoming webinar. And as I said, the plasma protocol is the 80CFB protocol, and if it's already at NECRAD, then there's no need to repeat it, although we'd like you to. You can never have too much blood these days. This field's moving so fast. The FDG component of this is a big way for us to understand the various types of neurodegeneration that may be occurring, that may inform on the etiologies that are there. And so this is optional, but we really encourage this for our impaired participants who have MCI or any form of dementia. And finally, the advanced MRI and disclosure pieces. So here's some other details of our inclusion criteria. We really want to focus on making this study inclusive as possible, and we want to have those conversations from the get-go. And we've got an incredible team in place led by Ozioma Okonkwo and Monica Rivera Mint and their teams. We have inclusion criteria that are pretty liberal. They need to have a NAC ID and be in the clinical course so that the data we collect can be linked up with the rest of their NAC data. Age 55, brain donor. And this is really one of the identities of our protocol, is that they're brain donors. Although this priority is superseded by whether they're a URG. If that's the case, we'll give them time to warm up to the idea of brain donation, and we'll be flexible there. We anticipate 40% unimpaired, 60% impaired. If they're unimpaired, we know there's plenty of unimpaired images in the scan database already. So we'd like to enrich this with people who have one or more risk factors for all-cause dementia. And if they're impaired, Alzheimer's disease needs to be among the considered plausible etiologic diagnoses. They can be amyloid negative based on a prior image. That's okay, but having this construct of Alzheimer's theoretically as in the mix should be one of the things to consider. All of these criteria should be traceable to their UDS forms, so that we can verify eligibility criteria. Here's a list of these risk factors for our cognitively unimpaired people. And thanks to those who've given some feedback on this. I won't take time to go through this here. It's in the protocol and you can review that. Exclusion criteria, there's no medications excluded. For those of you who use PI 2620, their licensing agreement is such that we're not supposed to use that tracer if the participant is an experimental amyloid or tau immunotherapy. And that's probably good advice across the board. If the person's already in a trial, they don't necessarily need to be in this study. Otherwise, major medical neuro illnesses that would affect participation would be excluded in your judgment at the site, and usual contraindications for PET and MR. And we will enable people to enroll who need a surrogate for the consenting process, and that's part of our consent and the protocol. The MR protocol, as Beth mentioned, the heart of it is the scan, adenine-4 T1 weighted volume, and flare. To this, we will add 15 minutes or so to do a multi-echo T2 star to derive high-res SWI for microbleeds, which will also allow for QSM to be quantified on that multi-echo T2 star. And Dr. Detra and others will give us an ASL sequence to be determined to assess cerebral metabolism. And we'll be doing an optional advanced T1 protocol with motion correction for perhaps more precise quantification of neurodegenerative signatures. And we'll be doing this, we'll be discussing this in another webinar. The blood protocol is the NECRAD ADCFB standard protocol. This is 30 mLs of blood. And you should, I think most of you are already doing this and sending 30 mLs to NECRAD already. If you're not doing that, please get familiar with their protocol and reach out to NECRAD to see how your site can qualify or get moving on that. Our planned assays so far include p-tau217 and these other usual suspects, as well as oligomeric alpha-synuclein and aggregated alpha-synuclein, which is, there's some prototypes available in blood, which we'll be experimenting with, and some other multiplex blood assays as well. We may be able to use a blood spot method as well, and that would be an exciting add-on if we can do that. Okay, to get to the some of the things you've been asking about, the heart of the matter here, I'm going to show you some numbers here that are based on the average, based on the site survey we did back in February of 2022. And we also assumed, using ADNI as a precedent, of a 30% indirect cost rate for the sites, which are all of you. We know that's not going to work across the board, and some sites just won't allow that. But we hope you will ask your grants office for that 30% IDC rate. And if it doesn't work, it doesn't work. It just means we'll be doing less imaging as a consortium, but we want to try to do that. So we will provide some startup costs, a one-time of 10,000 plus 30% is 13,000. Annually, we'll give you 10,000 plus 30%. So we'll give you 13,000 a year for community engagement and outreach events. We have personnel costs. This is one of the biggest barriers to getting a study started up and gaining momentum, is to have those personnel costs covered right up front. We are planning on having an outreach coordinator covered. We estimated about 50K a year for these positions, a little bit more than that, plus an assumption about fringe benefit rates. And we know these will differ from site to site, but this is roughly what we had allocated, 200,000 a year for salaries per site. For the procedures, I've graded these out, and I don't want you to pay too much attention to those numbers, but more so to these categories in the rows. We'll be paying you for an hour of MRI and compensating the participant for their time. And again, these numbers include that indirect cost rate within the dollar amount here. And we got these numbers based on the prior survey we did back in February of 2022. The actual cost for this will be negotiated with you. Similarly, there's a helicopter outside. Hopefully you can hear me. But similarly, for radioligands, we have budgeted for delivery of radioligands. We're using all the vendors, and I'll show you, well, several vendors, and I'll show you that on the next slide. But when the ligand is delivered and we have a contract, a central contract from Wisconsin, that bill will come from the radioligand company to us, and we'll take care of that. You won't even see the bill. For locally produced radioligands, depending on the dose and the agreement you have with the company, we'll be paying you for those, or you can request reimbursement for those. In some cases, this may come from the vendor, and then the vendor would pay us. It just depends on how your existing contracts are set up with some of these vendors. And again, the actual costs are going to be worked out with you for when you produce it locally. The ones that are delivered, we've already worked out those pricings with the companies, and we're getting a fairly good rate because of the leveraging power we have with all the ADRCs. Okay, here's just a brief status update on the PET protocol ligands that we're going to use. First of all, these ligands are all from the SCAN menu on the SCAN website. Please go there to see the radioligand options. From LMI for life molecular imaging, our contract with Florbetaben is in the final stages. The terms are all agreed to, and it's just getting signatures. For PI 2620, we've got a framework in place, and the master is getting signed now, and then the work order will be added to that. So that'll be a few weeks later. For Lantheus, which makes or licenses MK6240, one of our tau tracers, those terms have all been agreed to, and that's now going through final signature. The same for Enigma Mayur, who provides NAV 4694, which is one of our amyloid tracers. That's in the final stages. And Lilly is really stepping up to the plate here as well. they're going to allow you, if you're producing FTP, flutalsapyr, already, and you're authorized to do that, and you have an IND to do that, Lilly's going to allow you to keep doing that. And that's going to continue for the foreseeable future. We don't know when that'll end, but it's, for now, that's what we'll do. And we're also working on contracts with Lilly for delivery of florbetapir and flutalsapyr, FBP and FTP. So here's a little bit of how this works with the companies. Let me just take Cerveau as an example, which makes MK6240. There's a master service agreement. You could also think of this for florbetaben. We have a master service agreement in Wisconsin. Let's stick with Cerveau for MK6240. We will have a statement of work that will include all of the sites that want to use it and are in the network of delivery for MK6240. That'll just be attached to our master service agreement. Then we'll work with you to qualify you to order those doses and have those doses delivered to you and scheduled for you. And that radioligand company will then bill us in Wisconsin for that. If you are making doses locally using your local cyclotron and IND and so forth, you can also use that as an option for this to some degree. If you have a manufacturing agreement, for example, Cerveau, again, all the sites that are qualified to manufacture MK6240 have a manufacturing agreement with Cerveau. And so Cerveau would likely be treating you as a vendor to supply your local site with that dose. They will pay you, and then they will bill us for that dose. It's a little bit in the weeds of how this is going to work. For PIB, for those of you who are making PIB, you would simply attach, you already have your own IND for that. You would simply attach the CLARiTI protocol to your local IND, and you can go that route. So we'll work with you on all those various scenarios. It'll be exciting to see how everyone is doing this and try to make this work across all these centers. So disclosure has been one of the biggest issues that has generated a ton of discussion for us. And just want to give you a little update there, and I'll turn it over to Annalise to provide a little bit more detail. But first of all, we will be providing a central visual read. This will be done by Gil Rabinovich and company. And we would like more central readers to volunteer for this pool of central readers. And there's a little bit of effort that we'll supply to do that. So anyway, those visual reads will be coming from this central service. We want to develop a tau PET visual read. It's not there yet. So that will be coming later on. We won't do a central read for FDG. We won't do a central read for MRI, but we would like you to provide a central read of your MRI if it's applicable for that. We will do some central ratings on various things, microbleeds and white matter hyperintensities as a semi-quantification, but that's a research metric. And for the other disclosure pieces of this for amyloid and tau PET, I'd like to turn it over to Annalise if she's here.
I am.
Oh, nice. Thank you. I should have checked before I started all this.
I'm here.
Let me turn it over to you.
Okay, thanks, Sterling. So as Sterling and Beth have both mentioned, I think disclosure is something that is certainly on the minds of many of the centers. And this is a really great opportunity, not only to share resources, but to study disclosure. So generally speaking, we're going to strongly encourage disclosure of amyloid PET. So it should be offered to all, particularly those who are cognitively impaired. But as with all of our disclosure practices, as we're going to say later, we are not intending to be prescriptive about this. This is a site level decision. Many sites already have fairly advanced disclosure methods and protocols, so we are not superseding those. You can use the methods that we provide or your own local methods. We do ask that you modify the consent forms that we've produced as needed to reflect any local methods. In terms of disclosure of tau PET, again, if that's something that's already occurring at your site, that can continue. Again, we ask that you make modifications to the consent forms as applicable to reflect your local methods. We are also working on a centralized tau disclosure method and set of protocols as well. I wanted to just very briefly map out year by year what we're doing as a disclosure consultation team. So that team includes myself, Lindsay Clark, and Nate Chin from Wisconsin. We're also joined by Brad Dickerson and by Neelam Agarwal, who are both providing excellent mentorship around this. So in terms of year one activities and what we're doing now, we have been drafting over the course of the past several months a disclosure protocol and toolkit. The first big goal, and one that I hope you'll attend to, is that we'll be surveying all sites on your current disclosure practices. We feel that in order to produce a useful toolkit, we need to know what sites are already doing, what you're hoping to gain through disclosure, what tools might be needed, but also what you're doing that works well at your sites. We will also be consulting with and piloting this disclosure toolkit in this first year with 5 sites. I want to note that we are really intending to select sites who are, diverse in terms of the communities that they serve, the locations, geographically, expertise at each site, because we do want to have sort of a representative sample of the potential sites that might be participating. We'll then use that pilot site toolkit feedback, as well as feedback from the Inclusion Core led by Monica and Ozioma, to update this disclosure protocol and toolkit. So at the beginning of year 2, we will be producing kind of the finalized version of the training materials, a training certification program, as well as the toolkit and protocol itself. That will be available through NAC. And then in the following years, our role is really to help support disclosure practices at each of the sites. So we will be providing some in-person and virtual disclosure trainings, possibly tagged on some of the ADRC meetings if possible. We'll be evaluating the effectiveness of that training program, and then we'll be continuously and iteratively updating that disclosure toolkit and protocol based on preliminary feedback. And for sites that are interested in the disclosure science aspects of things, we will also be measuring outcomes following disclosure. Slide. So again, I want to reiterate, our goal is not at all to replace what sites are already doing in terms of disclosure if you have developed protocols, but we do want to support that. So our toolkits will include things like consent forms and language, decision supports, and informational brochures that are patient or participant-facing materials, many of which are developed through the Advisory Group on Risk Evidence Education in Dementia, or AGREED. We'll also develop some communication and report templates, training manuals, video scripts, etc. And again, we want to emphasize that strongly recommended would be disclosure of the amyloid PET results to cognitively impaired participants, and ideally to unimpaired participants as well. However, we do also have an optional, a set of disclosed results, including tau PET, other neuroimaging, etc. So we will provide some tools around those sorts of procedures as well. Happy to answer questions. I'll monitor the chat in the background.
Thank you, Annalise.
Sure.
Let me turn it over to Erin Chin, who's our overall administrative lead for the project, to talk about the site readiness checklist for CLARiTI.
Thanks, Sterling. Hi, everybody. My name is Erin Chen. I'm going to go over the site readiness checklist. This was something that Lori had included in the calendar invite. It's also available in a box folder that I will share on my next slide here. Just some things that your sites can start working on to ensure there's a fairly easy startup process for the CLARiTI project. This certainly isn't inclusive of everything that's likely at your site, but definitely a good start and things to consider as you get started at your site. So number one, making sure that we have appropriate contact information for everybody at your site that needs to be included on these webinars, any informational items that we send out. We're going to rely, if we don't have their information, we're going to rely on you all to share that widely with your teams as well. Make sure that your local Office of Research Services is ready for a quick turnaround of the subaward from NAC. That would just to help speed things up a little bit. And then if your site requires it, just make sure that you're working on seeding local IRB review to our single IRB WCG. So as Sterling mentioned earlier, we have that's currently in review with WCG. Again, in that box folder, there is the most recent version of the protocol and the appropriate consent forms. Start considering any site-specific information that will be included in the consent forms. You can see those listed there. Again, start working with your ORCORs to develop a robust recruitment plan for CLARiTI specific to participants from underrepresented populations. I know that Drs. Okonkwo and Rivera Mint are in the process of hiring a program manager for the Inclusion Corps, so that person will probably be onboarded within the next month, and will likely be in contact with your teams as well to start having some of those conversations. And then you can see on the next section there, the PET imaging, MRI imaging with scan protocol. So as Sterling and Beth mentioned, the survey that will be going out. So just making some of those decisions about what radioligands that your site will be using. Does your site continue to be scan qualified? Requesting local scanner time for CLARiTI. So any of those things that you would normally do for starting up a new project at your site. And then next on disclosure, Annalise just talked a little bit about that, but start thinking about what practices that you'd like to use at your site. And then again, biofluid collection. If your site is one of the sites that currently is involved in the ADCFB protocol, and if you're not getting on board with that, and then also collecting the kits from NCRAD as well. Sterling, next slide, please. And then I'm just going to introduce a few members of our CLARiTI admin team, because likely we'll all be in contact with you at some point in the near future. So you can see our email address there, clarity@medicine.wisc.edu. That will go to a general email inbox that Lori will manage, and she'll make sure that goes to the appropriate person. So again, my name's Erin. Lori, she's our administrative specialist. Hanja and Kelsey are our two project coordinators, so they will be the main points of contact for all of the sites in working through the CLARiTI project, doing any problem solving. Phoebe Frenetti, she is our communications specialist. She'll also be working with the team at NAC. And then we're still hiring for our lead regulatory coordinator. So I put a link there for the job posting. If anybody is interested, or if you know anybody interested, feel free to apply. And then thank you to Brittany and Brendan from the NAC team, who have created a really lovely website for us. So that is live as of today. So please go ahead and peruse the website. That'll be updated regularly as the site gets up and running, or excuse me, the study gets up and running. And then again, there's that box folder that we mentioned. You can go to that URL and you can find the protocol, consent forms, the site readiness checklist that I just went over, any logos if you need those for your local website. And then we'll also include the slide decks. And then after the webinar is done, Lori will upload the recording from this webinar as well. And again, we would like to have bi-monthly webinars, so be on the lookout for a calendar invite in February, and then we'll likely have one again in April, and we'll just continue that going forward, hopefully at least for the first year. And lastly, the site survey is coming your way, so that's going to include many of the items Sterling and Beth touched on, as well as Annalise to accommodate the disclosure questions. And lastly, I think this is it. Yeah, thanks, Sterling. We've got our wonderful logos. So as being part of this CLARiTI kickoff webinar, you can choose your favorite option. Thank you so much to Karen Thebes from Simplified Science Publishing. She works very closely with the NAC team and developed these really lovely logos for us. So Lori's gonna put a poll in Zoom. So hopefully that'll pop up in a second if it isn't already up. That'll be up for a couple minutes here. So take a look and please vote on your favorite, and we'll incorporate this into our documents, the website, and you can use it freely on your websites as well. And that's all for me. Thank you.
Thank you, Erin. I don't see the poll. Does anybody, is that happening?
Yeah, unfortunately, I just launched it and then it just disappeared. So I will make sure to
Do I need
send
to
that out
send?
after. My
Try it again.
apologies on that.
Otherwise, you
Yeah.
could, everybody could put it in the chat too.
You may
and-
be able to tell it to make a new poll just quickly here and just have a 1 or 2.
Sure.
Let me put that back up.
Yeah, can you put it back up?
Yeah.
Can y'all see that? Whoops.
Yeah, there we go. Thanks.
So option 1 has this sagittal view and looks like puzzles on the brain, which is very clever. And option 2 has more of a rendered, a top-down rendered brain, has a little more anatomic details on the left, and then a magnifying glass and a test tube inside of the right hemisphere. Both of these are very clever, and we can't decide. We need your help.
And unfortunately, I think I am just going to have you enter it into the chat.
A quick read on the chat is number 2 is winning.
All right, excellent. Well, we can compile those, but I think we can get started with the Q&A.
Yeah, there's a lot of good questions in the chat. I wonder for those of you guys who entered questions, if you're comfortable in just raising your hand or just asking the question, and we can call on you. We could do that. Otherwise, we can go through the chat one at a time. Yeah. Okay, Dr. James Noble. Yeah, Dr. Noble.
Thank you, thanks for putting together this program. I think we're eager to participate in it. One of the things we've encountered recently in some of our PET imaging comes down to the ability to use it for clinical purposes. And while we may be able to trust our research reads for kind of informing a diagnosis, our concern is that having a formal radiologist read on the chart is what's really critical for us to assure that somebody's not going to be, or that there's not going to be a pushback when it comes to insurance coverage if they end up getting on anti-amyloid therapies. And so I would encourage whatever we can do to have a pathway so that we can, if it's a local read or some sort of central read, again, so long as we have a clinic, radiologist read on the chart, I think in our view, that would really satisfy any concerns we have. And it may not even be that there's immediate pushback, but we just don't want somebody to get stuck with a fairly sizable bill considering the cost of these drugs down the line.
It's a great question. Some of these are going to be under IND, and that may or may not satisfy your local.
No, it would only be for the like FDA approved amyloid ligands is what I'm talking about. That's
Yep.
what we're.
Okay. We don't have funding in here for that kind of a clinical read of these images acquired under research. I will say that the team that Dr. Rabinovich is putting together they'll have to go through training on all of those things and have certification, and that will still be a research result that will return to you. But you can look at that and see if that works for the folks at Columbia. But otherwise, these images belong to you, and you could take them and get your own clinical read on it.
I think that's what.
Are they allowed to bill CMS for it and bill CLARiTI?
No, not for the read. That would have to
Just
be
for the scan.
not as we've structured it so far, John. It's
I think our plan would be that we would, if there's any fee or bill associated with it, it would be on the radiologist fee, and we'd have to figure out a way to have that covered.
okay.
Keep us out of jail.
Yeah, we want to stay out of jail. Thank you. All right. Well, if we can help you with that at Columbia, let us know. I think there was another.
Adam.
Adam.
Thanks, and I echo what's already been said. Congratulations on this amazing program, getting it off the ground. I'm a little worried about competition and recruitment and enrollment between what we're required to do under the new P30 RFA for the ADRCs, which is 24 ATN scans, and whatever we're on the hook for CLARiTI. I think when we filled out the surveys for this, I could get my timeline wrong, but it was before we knew of that requirement. So I don't know how we're going to resolve that, honestly. I mean that all the ADRCs are doing a lot of different types of studies, not just imaging. So I know at least for our ADRC, 50 amyloid, tau, and MRI scans per year might be a little bit tough for the resources that we have.
Yeah, thanks, Adam. That's basically what it would be. It would be you have 24 a year under the P30, and then if every site was an average enroller in CLARiTI, that'd be roughly another 24, 25 a year.
But those estimates were based on surveys right before we knew we had to do the 24, right?
Yeah, we did redo the survey just before we submitted. Once we knew the P30 requirement, and as we were resubmitting this, we redid just a short survey to say, is this still something that you guys want and need? And the feedback at the time was yes. And some of the sites aren't going to be able to do as many, of course. And I think your P30 should be the priority, get those 24 in, and we will try to support that as much as we can. By the way, for some of the sites that need that extra support, we'll be able to help you get your P30 requirement done if you don't already have a relationship with a radioligand vendor. We'd be happy to talk to you more about that too. Were there other thoughts you had on that, Beth or Nina?
I actually have a question about it, and I don't mean this in any bad way, but I'm just trying to more fully understand what the recruitment issues are. So the 24 required for the ADRC don't have restrictions on who the people are. So is it just like time on the scanner or scanning staff? Like what are the specific issues that make it difficult, or the actual recruitment of participants who are willing to do imaging?
For us, it's all of those things. You know, the PET center is used by a lot of groups. There are people going into trials. There are people getting clinical treatment with monoclonal antibodies now. And then, you know, 24 doesn't seem like a lot, but PET imaging, at least at Columbia, is a pretty complicated, very, very resource intensive in terms of personnel, in terms of transportation, in terms of the scheduling with the PET center, and in terms of competition with other studies. Maybe we're unique in that, but it's a lot of work, a ton of
No,
work.
probably not, but Sterling and Beth, correct me if I'm wrong, but I think there was a extensive survey trying to understand all those different factors. I'll let you guys weigh in on how to make that work, but I think that was definitely part of the discussion early on.
I mean, I think Adam's point is well taken. That, I mean, I think, Adam, you're saying that we might have to adjust our expectations based on the P30 requirements. You know, those initial estimates might be off of the average of 24. You know, that's what I think, estimated kind of roughly around, you know, per year before the P30 requirements. But I think the concept of being on the hook for CLARiTI, you know, these are just average numbers. And I think we expect some sites, you know, to do a couple scans per year for CLARiTI, and other sites to go beyond the anticipated average. You know, each site, you know, is going to be kind of different with these participant flows. So I think we're gonna have to see, I think, what happens here. But I mean, we have done these recent surveys where it does seem like there's still like a large amount of enthusiasm for additional scans beyond the P30 requirement.
Okay, thanks, Beth. That's helpful.
Maybe for the next meeting, we could do a quick poll, if it'll work for us, to see how many people think they can go beyond the expected number for CLARiTI, just to get a sense.
We can do that, Nina. Thank you.
Sterling, can I ask just one follow-up question on this? Brian Gold here from Kentucky. Thanks so much for putting this together. Great job. I'm wondering about, for those of us who have a scan compatible P30 protocol for scanning for our MRI and or PET, can we use the same subject's data essentially for who come from the P30 to be uploaded to CLARiTI if they have the scan compatible battery?
Well, that'll just happen anyway, because CLARiTI and SCAN are all together. And so if you did it under SCAN, you should just upload it as a SCAN set of scans, and the CLARiTIs will be separate. We want to make sure there's no even just the appearance of double dipping. So
Right.
if your stuff is already funded under that to acquire those scan images, just keep doing that. If you want to, on the other hand, extend your study by using the CLARiTI resources and give you more people into your project that you acquired them for SCAN, that's the perfect use of CLARiTI. They would just need to sign our consent form. Then that data then becomes yours to use as you wish.
I was thinking about more funding from the biomarker core, the P30 itself. And so, SCAN obviously had supplements, and those are over now. So we're not getting any money from SCAN anymore to do our work. That work would be coming from the P30. And so that's why I was wondering if it's separate, but could be used for the same subjects. But I think
in that case it is. This is basically serving that function to supply the money to upload those images to SCAN, but you'll just upload them to CLARiTI. They'll end up going to SCAN. SCAN will process them. It'll get treated the same way. It's just that we'll be able to get credit through CLARiTI for those dollars, and the dollars will come back to you through CLARiTI.
Cool.
Does that help?
Yes, thank you.
Craig had a question.
Yeah, it's really just a follow-up, on that as well. I mean, one of the great things about all of these efforts is the immense amount of overlap in the requirements and the techniques. But from my read of it, the P30s are sort of the minimum set of requirements, maybe followed by SCAN, and now here by CLARiTI. So as we're all trying to balance our enrollments, and yes, we cannot double dip. We need to make sure credit goes where it goes and everything. But if we can all strive to actually have a common set of procedures that more closely follows, CLARiTI here, even if CLARiTI isn't getting the credit, sorry to to you on that, but at least scientifically it would actually follow then the same data protocol. So making sure all of the scans and everything themselves are lining up, the blood and everything is lining up, etc. At least scientifically, we would end up with that final goal of wanting to have the biggest possible, most, complete, comparable sort of data set. As center directors and everything here, we can at least strive to do that.
That's our goal, and NAC is going to help us do that. And Sarah's here from NAC. Thank you, Sarah, and your team for supporting this. That's really what we want to have, is an integrated, highly leveraged, highly amplified set of data with this funding. Sarah, do you have any thoughts on that?
I mean, we're just so excited, and we're here to help answer questions as this moves forward. There is going to be a lot more data coming in. I mean, already over the past year, the amount of additional scan data that's come in has been enormous. This is just going to amplify that pipeline even further. So yeah, happy to answer specific questions, but I think we're on a great path here to get a lot more data in for the system and share it out.
Thanks, Sarah. Dr. Rovascali is our scientific officer from NIH, and her hand is up. You're on mute, Alessandra.
Yes, hi everybody. I just wanted to follow up on the issue raised by Adam about the competition for underrepresented minorities. And I think that there is an opportunity and there is an interest to open up the idea of underrepresented minority, not only as racial minority, but a broader concept of minorities and underrepresentation. So, don't be concerned only about racial ethnic groups, but open up and hopefully that more broad perspective will be helping to have less competition.
Thank you for that. Beth, do we have comments in the chat? What are your thoughts? Jim says he's giddy about starting the study and he's getting accused of voting twice.
I think there was a question about the INDs. So yeah, the IND, if it would go through Wisconsin, if you're using, if you're not doing local production, if you're doing local production, you'd be using your own IND. And then the CLARiTI would be an amendment to that. I think that answers that question. There was a question about the raters, if they need to be physicians or if PhDs are okay for the image interpretation team led by Gil. And I think right now the thought is that they'll all be physicians that will be selected. I know there's going to be plans to expand this initial team, so I don't know if there will be changes then. But for this the initial trained raters will need to be physicians. There was a question early on about CLIA certification for the plasma data. I don't know if Jeff is on, Jeff Dage, if you want to comment on that.
Will you say that question one more time? I missed the first part.
So there's just a question about for the blood test, whether they'll be CLIA certified, you know, and kind of how this might relate to disclosure.
Yeah, so right now, none of the blood tests are going to be run in a CLIA certified laboratory. And so they wouldn't be, you know, able to be used for disclosure at this time. But I think that is something we're interested in and we'll continue to look into as the project matures.
Thanks, Jeff.
Yeah, thank you.
I wanted to give a shout out to Ozioma Okonkwo, who's here. Ozioma, we didn't get to your slide. We'll do that in the next webinar and talk about our plan for diversity and recruitment. But is there anything you want to say or any context you want to provide now?
Thanks, Sterling. And again, it's great to see everyone here. And I know this is going to be a fantastic project, despite the heavy lift up front. I think Alessandra and both Nina have both shared thoughts, you know, on what the ideas are for inclusion for the study. You know, it's obvious, you know, that we need, you know, our ADC cohorts to be more inclusive, but also there's a lot of effort that needs to go into making that happen. And so, but with the expanded view of inclusion and representation that Alessandra and Nina both shared, we're hopeful that that is going to make this a bit more tractable for all of the sites. In Australia, you mentioned, you know, some of the funding that is being provided to the sites to make this feasible. So you have funds to conduct community outreach events at your local sites. You also have funds for a dedicated outreach specialist who is going to just be focused in helping your all cores meet your inclusion efforts. And then Monica and myself and the other members of our inclusion core are going to be reaching out to you guys over time to begin to strategize, to figure out what actually works best for your core and your site, because it's going to be different for each ADRC. So we want to partner with all of you on a one-on-one basis to make sure that we are doing this collaboratively to enable you meet the goals. 25% sounds lofty, but it's very attainable. And Monica and I have data to support this from other initiatives that we've worked on. But it's going to take all of us working as a team to make that happen. So that's about all I'm going to share now, and we hope to provide more information to all of you in subsequent webinars. Thanks, Sterling.
Thank you, Ozioma. for all the work you're doing, not just for this study, but for ADNI, HABS HD, and beyond, and probably many other things that you're working on. You're making a big impact. Thank you. Other questions?
There's one question about blood volumes to NICRAD, about accepting lesser volumes, you know, given the amount that's already being given. So I guess, yeah, one comment is if, you know, these participants, you've already given blood for these exact participants through another, you know, just as part of your normal flow or for another grant, you wouldn't have to repeat that for these participants if it's done within a year. So hopefully that will help, you know, somewhat with this concern. But I think you also might be asking kind of in general about giving lesser volumes. Which Nina responded that NICRAD is working on this issue. There was also a question earlier about using PIB. So yeah, that's okay. You can use PIB. That would obviously be through your you know, at your local site with your own IND.
That's one of the strengths of the Centers program is there's a lot of PIB being used, and that's the best in class amyloid tracer, and we've got a lot of it in the SCAN database already. And we want to keep encouraging you to use PIB if you're able to do so. And as Beth said, you just attach it to your own IND, and you would then be responsible for doing the IND monitoring associated with that. And we'll coordinate with you on study SAEs and AEs and those kinds of things, but this will be possible. Oh, and Sam's got a great idea to provide a letter that about the 30% indirect cost rate. And Colleen Roper, who's here, she's our research administrator. She's provided some guidance on that. It sounds great.
All right, if there's no further questions, we can end 30 seconds early. Thanks. Yeah, I want to thank again everybody for attending this and everybody's work, leading up to the grant and for going forward. Hopefully this is going to be a lot of fun and result in some amazing science throughout the ADRC program. And then we'll be sending out invitations for these webinars to continue. They'll be roughly every other month, and then we'll feature different cores within CLARiTI. And I'm sure probably for the next webinar, we'll also touch on study startup procedures as well, since we'll hopefully be just around the corner from actual data collection.
And can I have the last word?
Sure.
Which is to say thank you to Sterling and Beth. You guys have done a remarkable job. Nobody else could have done this. I'm serious. And I'm sure that everybody on this webinar here is amazed that you did it and is very excited. We certainly are. And the cure is around the corner.
Yeah, I was going to have the same last word. So thanks, John. And just on behalf of the ADRC program, thank you all. I know it was a very huge lift for everyone, and we really appreciate it, and great kickoff, and look forward to the new year. Thank you all.
Thank you,
I'm
Nina.
gonna second my colleagues.
Bye, everyone. Thanks, everyone.
Appreciate that, everybody. Thank you all. Happy holidays.